All right, today we're going to talk about crossing over. Now, crossing over, its end product is genetic diversity. So this occurs during meiosis, and actually during meiosis 1, during prophase of meiosis 1, where the chromosomes will actually trade off bits and pieces. And again, the end product is at the very end of all this, through meiosis 2, we'll end up with 4 genetically different haploid cells. So let's get a little overview again real quick. So right here, we're in interphase. We have a version of this chromosome from mom and a version of this chromosome from dad. DNA replication will occur during S phase, and now we'll have another copy there of that version of the chromosome from mom, and another copy here of that chromosome that came from dad. Now, what we have here is considered to be a homologous pair, right? So we have sister chromatids, and then we have a homologous pair. Now what's going to occur during meiosis 1 is we're going to get some trade-off of some of these genes. Sections of these chromosomes will trade off, and we can see that had occurred here. And then finally, during meiosis 2, when those chromosomes are separate from each other, then we end up with 4 haploid cells, and as we can see by the coloration, they are all genetically different in the genes that they carry, the version of the genes that they carry, which we call alleles, and we'll be using that term later on in the second semester. So we've got a couple new terms here, tetrads and synapses. Let's take it bit by bit here. First of all, again, this is the chromosome that came from mom, and this is the copy of that chromosome that was done during S phase, which is inside of interphase, remember that. And then we have here the version of the chromosome from dad, and the copy of that. So in most times during your the cell cycle, the life of the cell, it's only going to have one from mom and one from dad. It's only when we're going to do some go through meiosis, go through some cell division, which we're going to get these extra copies. So this here is a tetrad. So remember, tetra is a prefix that means 4. 1, 2, 3, 4. Over here, this is where the crossing over occurs. So these homologous pairs, they get real close to each other, and they form a tetrad. And 2 of them here are kind of looks like they're crossing, they're crossing over. And what ends up happening is this piece here will be split off and go to this chromosome, and we can see that this piece here was split off and is now going to this chromosome, and we have our end product here. We have 4 genetically different chromosomes, which will go to the gametes, the sex cells. To look at a little bit more in depth, here we go. So we have meiosis 1 and meiosis 2 here. And like I said, this occurs in prophase 1. So the chromosomes condense and the nuclear envelope breaks down. Crossing over occurs. Now we move on, metaphase 1. Pairs of homologous chromosomes move to the equator of the cell. Nothing different like we've seen in all other metaphase. Anaphase, the homologous chromosomes move to the opposite poles of the cell. Same thing that always happens in anaphase, whether it's in meiosis or mitosis. And in telophase 1 and cytokinesis, we have the chromosomes gathering at the poles of the cells and the cytoplasm also dividing. Now, if you look at prophase 2, there is no crossing over like what happened in prophase 1. Crossing over only happens one time, and that's in prophase 1. And at the end, as we see, go through the entire process, we can see our 4 genetically different haploid cells being developed in telophase 2 and cytokinesis. Okay, so again, why? Well, again, it all comes down to diversity. This creates diversity. Why is diversity good? Well, let's throw some facts at you here. Each new haploid cell is genetically different. I keep saying that over and over again because it's important to remember. Now this creates almost unlimited possibilities and variation that we can see within a species. That's why we see people they don't always look alike. We don't all look alike. There are differences between us all. But why is that good? Well, diversity allows us to adapt to a changing environment. So if an environment changes, let's say there's an external stimulus that comes about that applies a pressure, natural selection, we can say, which we'll get into on the next unit. But What that allows is that perhaps there are individuals of the population that have this adaptation that allows them to survive that stimulus a little bit better. And genetic diversity gave that person that ability. If we were all exactly the same, and we were all clones of each other, and let's say one disease came into the population, that one disease could possibly wipe everyone out because no one would have a defense. But possibly through genetic diversity, 1 or 2 or 3 individuals of that population could possibly survive. So that's good for the species. All right, so that's crossing over. It's pretty simple. It occurs in prophase 1 during meiosis 1, and we have a switching of genes from one chromosome to another, and the end product gives us 4 genetically different haploid cells. If you have any questions, let me know in class, or hit me up in the comments. Stay in school.